Good morning, students. Hello, everyone. Hi, 5S2, are you there? Okay, I hope you're bright and early and all up already for today's class. Okay, today I have an interesting lesson for you, and it is regarding population ecology. So we're going to do a bit of calculation, how to determine the population of a certain species in the ecosystem. Okay, so let me look at the chat. Okay, good morning, Xin Hui, Min Yin, Kiet Yi, Jia Wei, and Kersin. Good morning. Okay, I hope I can. Okay, is the sound system, is audio clear? Okay, so let me add to screen. Okay, let me look at the share screen. Okay, are you able to hear me? Fine. I think okay. I can see the slide already, so I think no problem. Okay, let me start today's lesson. Okay, for those that come in later, you can rewind back and just have a look at the beginning part. Okay, now today we're going to talk about population ecology. So in your textbook, okay, this is the part that I want to cover. Okay, let me look at some more comments here. Okay, yes, all right, good. Thank you very much for your feedback. Okay, Yuning, Xin Min, and Xin Hui. Now, so population ecology, as you learned earlier, the definition of population is a population refers to the same type, is a group of the same type of organism or species staying together in the same area. So what we need here is the word same species. So when you talk about a population, you are referring of all the individuals of the same type. So like I say, if a population of the of students in the school, that means you're counting for the students only. Okay, if you're asking for the population of teachers in the school, they're only asking for the number of teachers in the school. So population is only for one type. And you do not mix different types of organism or animals and plants together as a population. So population is just talking about the same type only, and it must be staying in the same area or the same habitat. Okay, so the keywords here, population is referring to a group of organisms of the same type or same species staying in the same area. So today we're going to learn about population ecology. Okay, ecology is a field in biology which studies on the interaction between the what we call the components, the biotic components and the abiotic components with the environment. Okay, so it's something like we learn about ecosystem, right? When we learn ecosystem, we are learning actually about ecology. So ecology is the field of science which investigates the relationship, the field of biology, which investigates the relationship between this organism, okay, staying in this area, in this ecosystem. What is the interaction between this animal and another animal? Are they competing or are they fighting? What do they want? Do they want the same resources? Okay, or is this a prey-predator relationship? A eats B, okay, B eats C. So what kind of relationship is it? And also with the environment. So when we study ecology, we look at all these things. Okay, so let's look at some terms here. Okay, so talk about population is actually talking about how big is the size, how is the animal, the number, and where do they stay? Are they spread apart and so on? So population is a group of organisms of the same species living in the same area. Okay, when you talk about size, population size, okay, look at the keyword population size. You're asking for the number. Okay, how many organisms are staying there? So when the question asks you what is the population size of chicken in the barn, okay, they're asking about how many chickens are there. In other words, simple language. When they ask for population size, they ask you about how many. That's what it means. Okay. Then population density. Density in Huayi, Mandarin, we say 密度. That means how close are they staying together? Are they in a very small area? Is it 1,000 there? That's considered very high density. They're very close together. The living space is so little, but there are too many animals staying together at the same place. That is called density, high density. If you say density is low, that means in the same area, you don't have many. That means they are spread apart. They are spread away from each other. Okay, that's called low density. So when you talk about density, you must divide by the area. Divide by unit area. So it's always per meter square. Okay, how many individuals or how many chicken per meter square. That means in 1 meter area, there should be how many chicken if you spread all of it out. Okay, that is what it means by the density. So there's population, you can talk about the size, that means how many, don't care the area. But if density, you must take the size, how many, divide by the area. Usually we use meter square. If for very small organism, we may use cm square lah. Okay, right. Okay, so far everything okay. Now, population abundance is referring to both. If they ask you abundance, they want to know about what is the population abundance of this organism. That means they want to know the size. They also want to know the density. Okay, so size and density is talking about the abundance. All right, okay, so we got some terms cleared out. So population ecology is the study of the measurement of a population size. So when you study about population ecology, you're going to study in that area there how many of the organism you want to study stays there, the size, and also what are the factors that affect the size. Okay, now for example, why is this organism staying in this area, this habitat A, so many, but in habitat B, why so few? The same organism. Why in B habitat B is so few? There must be certain reason. Okay, so what are the reasons that affect whether this animal live a lot in this area or live very little in another area? The factors will be actually depending on two components, whether you have abiotic factor or biotic factor. So abiotic factor is like temperature, maybe the area is very hot, okay, not too suitable for the animal or for the plant. Or maybe the pH is too acidic, so it's not suitable for the plant. So pH is more for the, talking about more for plants, okay, the soil pH or the water pH. Then the water source, maybe the area there not much water, that's why they cannot grow very much, okay. So these are the abiotic factors which affect the population of an organism in the area. Why here a lot? Why over there very little? Because not enough food, not enough water. Okay, that is abiotic. Then you talk about biotic. Biotic means the interaction with the living things that maybe there are other animals who they are living together with other animals. Maybe there's competition, maybe interspecific, intraspecific. Okay, they fight for the same thing, so they fight, and then of course some of them die off. So that's why the population is not so much there. It's not so high population. Maybe there's a lot of competition going on there. Okay, or maybe there's parasitism. Maybe there is infection of certain these worms inside this species, right? That's why they don't grow very well, or they do not multiply. They don't have a lot of these animals staying there because maybe they die of sickness. Okay, parasitism will cause sickness, and also maybe predation. Predation means one animal eats another animal. So maybe they stay together with a very ferocious predator. So the predator will keep on eating up their numbers. So that's why the number will dwindle, the number become less. Okay, so these are abiotic and biotic factors which affect the population. Okay, other factors would be like birth rate, how fast they reproduce, and death rate, how fast they die off. Okay, the new generation will be born and then the old generation will die off. That's called the birth and the death rate. Okay, death rate, another name is mortality rate. Okay, just a little new word. Mortality rate also is the same thing as death rate. That means how fast are they dying. So they will die faster if there's illnesses, if there's sickness, or there's a pandemic, or there's an epidemic. Epidemic and pandemic is the difference. Epidemic is just for the area only. They call it epidemic. Like for COVID-19, it's called pandemic. Pandemic means because it's spread all over the world, everywhere. There's no country which is not affected by this COVID-19. So we call it pandemic. It's all over the whole world. Okay, but if epidemic is only for that certain area only, let's say epidemic in that certain country, other countries not affected, then you call it epidemic. But if it's so widespread like COVID-19, we call it a pandemic. Okay, so the rate of transferring out, maybe some organisms, they will move out. We call it emigration. Emigration, like in Mandarin we say 移民, it means they go out of the area. They go out for certain reasons. Maybe they lost the fight when they have the competition. Okay, they lose to another male which is more dominant. So they will transfer out because they know that they cannot get the place that they want, the cave they want. So they move away. Or they move away because of that place no food. So they move away. Okay, berpindah. Immigration is coming in. So maybe got new new organisms from another area just move into the neighborhood. Okay, this is immigration. They come in, so they transfer in or they transfer out. This also affects the population. When the immigration rate is higher than emigration rate, that means the rate of new organism coming in is higher than the rate of organism going out, then of course the population will increase. If the other way around, emigration rate is higher than immigration rate, that means those going out is more than those coming in, then the population rate will, the size will decrease. Okay, now let's look at, before we go to how to calculate the population, let's look at what are the factors affecting the population distribution of plants. So we look at plants now. What are the things can affect whether the plant can grow very well or whether the number is a lot over there? Depends on what, okay? So just look at what the plant needs. So the plant needs suitable temperature. So the optimum growth for plant growth is actually between 25 to 30. A lot of people, a lot of students always make the mistake of thinking it's 37 degrees Celsius, which is wrong. For plants, the optimal temperature is not 37. 37 is for animals. Just like our human body, our temperature is 37 degrees Celsius. That is the optimal, that means the best. It's the best temperature for our enzymes to work. Okay? And for plants, it is not 37. It is between 25 to 30. It's around roughly 27. So you can just imagine 10 degrees lower. All right, okay? So now why does temperature affect the distribution? Okay, good morning, Alpha Danish. Okay, good morning. Yeah, thank you very much for your sokongan, Alpha. Thank you very much. Okay, I know you're very rajin join our biology class on Monday, 9:30. Okay, thanks for joining my class this morning. Okay, so now temperature affects, because always tie in with enzyme. When you talk about why temperature affects the organism, don't care whether it's plant or animal, it's because of the enzyme. As you know earlier, enzyme, they will react only when they have a certain optimal temperature. If it's too high, all right, if it's too high, temperature which is too high, they will denature, or you can say destroy, okay? But denature is still the best word, okay, to be destroyed. Denature is the best word for you to use. In BM, we say nya asli. Okay, the high temperature, remember? So when the enzymes do not work very well already because the shape has changed and the substrate is still the same shape, it cannot fit into the configuration, the active site. It cannot fit inside there. So when it cannot fit inside there, the enzyme cannot work to increase the rate of converting the substrate into the product. Okay, so it doesn't work already. So therefore, the process of digestion, the process of respiration, everything slows down. So it stunts the growth. That means slow down, right? The growth of plants. Increase the rate of water evaporation by transpiration, because it's hot. Temperature high means water evaporate faster. So water will lose, okay, will be lost. They also slow down the rate of photosynthesis. Okay, now for this one, if the temperature is low, okay, so I have many students always give a wrong answer. They say low, they say denature. So when it's low, never say denature. Denature only happens at high temperature. When the temperature is low, you say the enzyme is inactive. Good enough for you to say the enzyme is inactive. So when it's inactive, means the enzyme do not work. They will slow down the biochemical reaction. Okay, so remember, high temperature is for denaturing the enzyme. Low temperature, you say the enzymes becomes inactive. Okay, right. Okay, thank you, Alpha. Thank you, Alpha Danish. Right. Next, water. Okay, now water, of course, water is very important for all life, all cells. You need to have water because water will is the medium for biochemical reactions. You learned in Form 4 before the importance of water. Without water, a lot of chemicals, they don't get dissolved. They don't get dissolved, they do not act. They do not have the function of showing the characteristics. So you need it for enzyme, right? To break down substances, you need enzymes to speed up. You also need water for the chemicals to work. Photosynthesis, photosynthesis, one of the raw materials of making food is actually carbon dioxide and water. So without water, you can't carry out photosynthesis. Transportation, transporting the nutrients in the xylem and phloem, you need water. And also for turgidity, to support, to make sure the cell is not flaccid so that the plant can stand up straight, okay? It does not become wilted. So Now, dry areas, certain areas which are dry, like the desert, right, in Sahara Desert and Gobi Desert, all this, you have limited resource of water, so the plant will not grow very well over there. And only certain plants which have adaptation can grow there. For example, the xerophytes, we learned in chapter 7, special adaptation, right, sunken stomata, succulent leaves, and so on. Okay, so they will have low population dense distribution in places with less water, all right, and also low density of plants. Now, tundra are those areas that are very cold, very near the northern part of Russia, and also like this Arctic, all right, Canada, the northern part here. These are very cold areas. They're always with ice, so this is tundra. Okay, light, as you know, light is needed for photosynthesis. Definitely, it goes to show that it goes logical to say if you do not have enough light, you cannot have enough photosynthesis, so the plant will not grow very well. So plants always look for sunlight. When it's talking about green plants, not talking about fungi. Fungi, they don't want light because they don't have chlorophyll. Okay, so areas which have little light will have a lower number of plants. It makes a lot of sense. We're talking about green plants. Green plants, okay, not just any plant. Because if fungus, it doesn't affect. Okay, pH of the soil. Soil pH is important for nutrient absorption. So it cannot be too acidic, it cannot be too alkaline, because certain plants prefer acidic or alkaline condition. For yeast, they prefer more acidic condition. For bacteria, they prefer more alkaline condition. Okay, so they will lower the absorption rate of nutrients, so they do not grow very well. Okay, lastly, mineral salt content. So it has to do with how much salt content in the soil. If it's very, very salty, that means very high content of salt, it is hypertonic condition. The water from inside the root will actually diffuse out. Okay, so the plant will be losing water. So it is because of the process of osmosis. So that's why many plants are not able to grow in very salty soil, except for the mangrove swamp, which we have seen last week. Okay, our lesson last week, last Saturday. So mangrove swamps have special adaptation, even though the soil is a high salt content, but inside the root is even more higher salt. So the water still can go in. But other plants, you don't have the adaptation. Water will come out of the root. That means it will lose water. Once it lose water, then very soon it will wilt, and then slowly it will die off. Okay, so high salt content in the soil will cause the plant to lose water through osmosis. So for normal plants, you don't put too much budget, don't too much this what they call the fertilizer. It will increase the salt content in the soil. Water will come out, and this is a very common question. Always they ask you this K-part question: why you cannot put too much fertilizer in the plant? Okay, remember that is Form 4 already. Form 4 work. Okay, so minerals are needed. They also need to absorb mineral, all right, but not too much. Too much, you will have this hypertonic condition. So phosphorus will be used to form phospholipid to make the cell membrane. Okay, this is for plants. So these are the conditions which affect whether the plant can stay there, whether it's a lot or very little. Okay, what about animals? What are the conditions that will affect whether the animal stay there, or a lot, is there a lot animals stay there, or very little, very few animals stay there? So it has less conditions here. First of all, will be, of course, temperature, right? Too hot, animals cannot survive. So they need to look for a place to stay. They go and burrow into the soil, or they go and look for a cave. Okay, so high temperatures would be less inhabited. Too hot, right? Too hot, water from a lot of what they call water will be evaporated. So you will find that you will not enough water, right? Now water animals are concentrated in waters that have enough places that have enough water. Okay, makes sense, right? So they will stay if there's a river, there's a lake nearby. You'll find that generally the population will be higher. Because for drinking and also cooling the body, like animals like hippopotamus, they need to be near a lake. They always will wallow in the mud there, right, to cool down the body temperature. Breeding site, whether there's a suitable place for them to reproduce. Okay, usually a cave or a place for them to make their nest so they can look after their offspring. So if you have it there, then the higher chances of the population to be more. Then food supply, this is important. See whether the place has got food for them to eat or not, right? If there's no grass, there is no vegetation, there is no animal as their food, definitely they cannot grow there. Because animals, they're all heterotrophs. They cannot make their own food. They have to eat food from somewhere else. Okay, so these are, and also for this is for a factor, what we call abiotic factor. For biotic factor, you have mates also. All right, so you have mates. There are a lot of female there. All right, the male will try to. It's a natural instinct. They will mate with the female. So this is a biotic factor. If there is mates there, right, of course they will thrive. They have a reproduction will happen at a higher rate lah. Okay, now so we've done with all this. This is not so important, right? But more is important. What is more important? I want to teach you how to take sample, how to study in that area. We want to study how many animals are there or how many plants are there. So we need to count, right? Correctly? Correct. Now, the first thing we need to do is to count. Now, counting is not a problem. It is very little. For example, you go to your garden, right? You go to your garden outside your house. Yeah, your garden, maybe you have pokok semalu there, all right? The Mimosa pudica. And maybe your garden is not very small. Your garden may be 10 meter by 10 meter, let's say, all right? 10 meter is not very small. Just about 10 meters, not 10 meters, quite big. So, so maybe 5 meter by 5 meters, okay? A small little patch there. No problem, you can go to a garden and you can count every pokok semalu you can see. You can find out, just find and count how many. You'll finish maybe in the 5 minutes time, okay? But if your garden is very big, you have a one whole big field or football field, right? It's not garden, let's say you want to investigate the pokok semalu at that football field that has not been used for a long time. You want to count the number of pokok semalu in that football field there. Football field, as you know, is very big. You cannot be walking all over the field, the football field, to count your pokok semalu because it is taking too much effort. Okay, it's so big area and maybe there's a lot of pokok semalu. You count until so you give up. So we cannot be physically counting if the area is very big. So what we do is we do an estimation. So this is now we're talking about a population ecology. An estimation by using a statistical method. Statistic. This is about statistic. We take sample. We call this sampling. Sampling is a representation. When you take sampling, it is representing of the whole thing. You don't have to count every plant there. But if you do it correctly, using the sampling technique is correctly, it will give you a rough estimation and a quite accurate one of actually how many are there without really counting. Because counting is too time consuming, especially with the place is so big, right? So we do a sampling to give you an estimation, will give you an answer that is quite close to the actual number without really counting one by one. Okay, so that's why we do sampling. So sampling, how do you, they are in a place where animals or plants stay, the population distribution could be not evenly distributed. For example, it could be clump. Let's say you go to a certain field there. This is a field, look at the first one. You could find maybe a lot of pokok semalu in one area there. Then a cluster of pokok semalu another place. Then another cluster. Means this is not evenly distributed. It is not so even. You will find places that will have, some places don't have. So this is clump. Clump means it groups together. So maybe that area got more water. That's a wrong reason. Or maybe that soil there is richer. That's why the pokok semalu will grow a lot at one place there. While the other areas yang kosong one, empty one, maybe that area, the soil is drier, or maybe the soil is too acidic or too alkaline. Therefore, these pokok semalu don't want to grow there. Okay, so they were from groups. This clump one, the pattern, we're talking about pattern, right? How they grow. So they could be in groups, they are separated, and they are uneven distribution. They are not even because here a lot, there nothing. Okay, now another one is random. Random means there's no pattern there. Here got, there got, you don't see a clump also. So it's randomly distributed. So it's a free pattern. It's also possible, right? You can get here and there everywhere your pokok semalu, right? Then next one, uniform. This one is quite rare unless human plant the plant uniformly you put 1 meter, 1 meter, 1 meter distance okay. If not usually you don't get uniform So these organisms are distributed all over, all right. And then you get the distance between one and the other is more or less the same. So this is random. Usually you will get this and this in nature, right? A uniform distribution is quite rare okay, because if uniform is more like man-made, all right, purposely put the seed here, and then you find it's all nicely in a box there, the distance. Okay, so now we look at how to get your sample. How can we get the estimation of the population without actually counting one by one, which is going to be very time consuming, take a long time, especially when there's so many. So we're going to do estimation method. Okay, estimation method is actually sampling. So the method is called sampling technique. Okay, so sampling technique is done to study the population size. That means you want to know this organism I want to study is how many in that area there. All right, you want to know how many, and also distribution. The population size and distribution actually same. Distribution. All right, so how do we do it? We collect, okay, collect sample. We count, important for you to count. We collect, we must take sample. We count after that. Then we make observation, write down. Then we put all these figures in a formula. So I'm going to introduce the formula to you later. Okay, so sampling can be done two ways. How to take sample? How you want to take a sample from your area that you want to study? Do I go and purposely look for the pokok semalu at one area, or do I do randomly? Okay, so there are two ways. For you to do sampling, there are two methods. You can do randomly or you can do systematically. Okay, what's randomly? Randomly is like here. All right. First of all, you need to take a sample. You must make a frame. So for animals, for plants, usually you have to make a frame. We call it a quadrat. The frame is called a quadrat. All right. The quadrat you can make from kayu. All right. You can make from wood. You can make from metal. As long as a frame, and you must know the area. The quadrat important for you to fix the area. All right. For here, example is 1 meter square. Okay, this is 1 meter square. That means 1 meter by 1 meter. 1 times 1 is 1 meter square. So inside there, you can put smaller, what you call, smaller lines or boxes by drawing, by pulling string, right? By putting string there. So this is for plants. For quadrat sampling, we do for plants, not for animals. Usually not for animals, because the plants cannot move. When you put your quadrat there, right? Once you put your quadrat there, you can see, you can count how many plants that you want to investigate. Let's say pokok semalu, how many you see inside that quadrat. Okay, you can count, and they don't run away. That's a good thing. When you do quadrat sampling, you put the frame there, the animal or the plant is still there. It doesn't move away. So you just count how many. Okay, now for the first method of random sampling. Random sampling is like this. You put your quadrat anywhere you like, without certain method. So let's say this is the first quadrat. All right, you put there, right? So this is random. We're talking about random first. So first there, you put there, and then you count. On inside that quadrat got how many plants that you want. Let's say your pokok semalu, you're doing pokok semalu investigation, a study. So you count how many pokok semalu. Don't count the rest. We don't want the rest of the other species. You only want pokok semalu. So here you got 1, 2, and 3. Now, if your plant is inside, you should consider it here as count. Usually it's more than half and more. Okay, half you also count. So I'm not too sure why you don't need to count here. Looks like it's more than half. So usually when it's more than half, you take the convention of counting it. If it's less than half, okay, you don't count it. So here you find that there's 4, right? So the first quadrat you have 4, so you write down, right? You have to measure, and you have to write down in your table. Then you take your quadrat, you can use the same quadrat, you don't have to make 10 quadrat. You put it another area, let's say here. The same quadrat, you put it here. Randomly, that means you just simply throw the quadrat. Just simply throw a quadrat and see where it lands, right? You go there and you count. So here, the second quadrat, right? It's the same quadrat you can use again, all right? This is now 5. Okay, you repeat. You throw it another area. You get the third quadrat, you get 3. Okay, you do another one. You get 1 here. This is quadrat number 4. So you repeat. Now, how many times do you do it? So usually for quadrat sampling, you usually do more if you want to have a more accurate result. So it depends on how lazy you want to be or how hardworking you want to be. If you really want your answer to be very accurate, that means actually there are how many pokok semalu you really want to find out. You want to make the answer really, you want to get answer that's really accurate. It means very near to the actual number. Then you got to throw more times. You do the sampling, you do more. The more sampling you take, the answer will be more accurate. So minimum people do 10 right? Minimum 10. Then if you want to do 20, 30, 40, doesn't matter. It can overlap by the same. Once you take it away, it's considered like a new area. You can just like a 2 and a 3 now. Now your 5 can be here. It doesn't matter. You can overlap the area, right? You go through many times. You do it, you get more samples. The more sample you get, the more accurate your answer to be for the estimation. Okay, so you write down every time you get how many pokok semalu, you write down. And if you land on the area don't have, you put it at 0. So you call randomly, you do not let your mind be affected. Don't purposely throw it because you see the semalu there. That is wrong. It's cheating. You don't throw, don't care whether you have or you don't have pokok semalu, just throw randomly on it. All right, don't care where it lands. You just count. Got semalu, never mind. Don't have, also never mind. Just randomly throw. Okay, so that's one method, randomly one. Okay, now another systematic. Systematic is there is a system where you put your quadrat. Let's say this is your field. Your field is this big. Okay, and then my quadrat is, let's say, 1 meter by 1 meter. So I put my first quadrat here, right? Then after that, I measure a distance of 1 meter. Then I put another one. I do the second one. Then I measure 1 meter. Then I do another one. Okay, so this is distance is fixed. So another one, 1 meter. In between here, I do another one, my number 4. Then here, number 5. Here, number 6. So you go through the whole area by putting your quadrat systematically. Okay, make sure the distance is roughly the same. You measure the distance, then you put another one. Measure distance, put another one. This is called systematic. Okay, more work more work because you have to measure the distance and so on. Okay, so that systematic also works. So either use random or systematic, but don't use your method of whether I want to put there because I see a lot of pokok semalu there. You purposely put there, you will affect your answer. What it means is your answer will get more than necessary. Or on the other hand, let's say I'm very lazy to count. I purposely put in the area where I know very little pokok semalu. I purposely put kosong punya area. Kosong means I don't need to count, isn't it? Then your answer will come out as kosong. Means in the end, you find your population, your estimation will be very low. Okay, so don't let all these factors affect you. Simply throw random or put systematically, and you will find that your answer will be a bit more accurate. Okay, and increase the number of sampling. So now, how big is a quadrat? Right, can be how big? Depends on what kind of plant that you are doing investigation on. If a pokok is very small, you don't need to have 1 meter size quadrat. Okay, for example, for mosses, lumut, right? Lumut, you don't need to have 1 meter so big because the moss is, it covers a small area. You can have 10 cm by 10 cm. So 10 cm is actually very small. You can make a frame 10 cm, all right? You use plastic or use kayu or that. Then you just put a fork. Or you can put on, you call it the plastic. You draw a square on a plastic sheet and then you put this onto the moss and then you count the area. Okay, you count the area that got moss. See the area got moss. This is for coverage. So you can use it for size is more for small plants. Okay, lichen and mosses. These are the ones that on the rocks or on the tree barks. Kulit kayu. Sometimes you get the lumut. You use this small little plastic sheet, just like a plastic sheet. Just like a piece of paper, but it's plastic. You just draw 10 by 10. You just put it on the thing and just count. Okay. Or for grasses, you usually 1 meter by 1 meter. So you put your kayu 1 meter long, and then you paku put your nail there, you paku and make a square like that. Then you bring it to the field. You bring it to the field and you just put anywhere you want or systematically. Okay, then you just measure count. Now for bigger trees, 1 meter sometimes not suitable because the tree is big. 1 meter you maybe barely have 1, or sometimes you don't even the tree so cannot masuk there. Cannot masuk your 1 meter. So you maybe 5 by 5. And for in the forest, if you want to go and do your study on pokok meranti, pokok jati, pokok cengau. Now these are the huge tree we have in our tropical rainforest in Malaysia. You cannot be 1 by 1 because your tree trunk is already so big. Okay, your tree trunk is definitely so big. So what you do is maybe 10 by 10. But 10 by 10, how to make? 10 by 10, 1 meter is already so long. 1 meter is like your arm span. It's already like 1 meter, right? How to make 10 meter and make the kayu, the quadrat, and bring into the forest? Okay, you don't do that. For very big quadrat, you don't make it and bring it there. What you do is you go to the place when you want to do your investigation. You make your quadrat there. Alright, this quadrat no need to use kayu. You just put a what they call you put a kayu onto the ground, and then you tie a string 10 meter long, and then over there you put another kayu. Now this is your length already. So that means like this. Here is your kayu here. All right, you're in the forest. You're in the forest. You put a kayu here, and then you are here, right? You're here. Then you go and measure 10 meters. Then you put another kayu. This is 10 meters. And then here you put your string. You go again, measure another 10 meters. Here another kayu here. Then here 10 meters. 10 meters. You already make a quadrat on site. Okay. And then here is your kayu, pokok lah. Maybe in here you pokok kayu cengau or meranti. Inside there 10 by 10 meter, I get 3 species. Okay, this is for big trees that you want to take sampling. Okay, this is called sampling for these big trees. So you can place it or you can throw randomly. Now, usually you do 10 quadrats, okay, to get an accurate result. The more, the better. Always remember, this is statistical rule. We learn about statistics later in form 6, if you do maths. The more sampling you take, your answer will be definitely more accurate. So statistics, okay, sampling. Now, how do you count with all your results? How are you going to get your number? Okay, so now we're going to learn how to do the calculation. Okay, from the this value that you get, there are 3 things you can measure. Whether the question asks you for density, what is the density of your pokok semalu, or what is the frequency of your pokok semalu, or what is the percentage coverage. This is usually not for pokok. Pokok can count. This is usually for grass or for mosses, or for trees that you find that the plant you cannot really separate one plant with another plant. So we just see the area and the coverage, how much is covered by the area. Okay, we go one by one first. All right, okay, I think no questions so far. No questions so far. Okay, so let's carry on now. Now, first of all, you need to know, let's say a question asks you, what is the density? Given the readings here. Okay, first quadrat, when you throw your quadrat the first time, you get 5 pokok inside there. Individual is 5. The second time you take away, you throw another place, you get 4. Okay. The third time you throw your quadrat, you get 3. The fourth time you throw, you get nothing. Kosong. Your pokok semalu not inside there. Okay. Then the fifth time you throw, you get 3. So you can see you have 5 figures here. How to find the density? That means how close is your pokok semalu in the area? How near are they? How many pokok per meter persegi? Okay, how per meter area, per meter square? Okay, answer, you do this. You first of all, density equals to count all the number of species in all the quadrat. That means you add all this. 5 plus 4 plus 3 plus 0 plus 3. So here 5 plus 4 plus 3 plus 0 plus 3. Add up. Okay, add up all. Kosong don't have, you can leave it out. Okay, but make sure you add up all. Divided by the total number of quadrat used. So you can see how many quadrat did we use? Okay, we use 1, 2, 3, 4, 5. Okay, so we use 5 quadrats. So divided by 5. And what is the area of 1 quadrat? So the area of 1 quadrat is always given to you. So they say that the area of quadrat is 1 of the quadrat, each quadrat is 1 meter square. So you times 1 meter square. Okay, and we push over to this side. So if you add here is 9 plus 3 is 12, 12 plus 3 is 15. Okay, 50 divided by 5 times 1 is 5. Now the make sure you remember the unit. Your meter square is still there. On top here is number. How many? You call it individual. How many plants? There are 15 plants in an area of 5 meters square. Okay, 15 pokok semalu are found in an area. You took the sampling of 5 meters square area. So you divide, you're going to get 3 individuals per meter square. Or you can put like this. There are many ways you can put here meter square, or you can put 3 per meter square. That means in 1 meter area, roughly there are 3. That means even out lah. If you evenly distributed all your pokok semalu, there are about 1 meter square, you can find 3 plants. Okay, so here remember, your answer for density is always per meter square. Or we say individual per meter square. Always got area 1 for density. always with area. Okay, so this is not difficult, very easy for you to do. Simple mathematics. Sometimes you don't even need a calculator also. Okay, next one, frequency. What's frequency? Frequency refers to how many times did you find the plant in your quadrat. How often do you get it? When you throw it, do you get every time also kena your plant? Or are there plant times when you throw your quadrat but no plant there? Okay, so this is called the frequency. Now, first, what you need to do, frequency is in percentage. Okay, we times percentage. How many percent of your sampling you get your plant? Okay, so here, let's look at the example. First quadrat, you get 2 plants. Second quadrat, you get 3. Third quadrat, you get 1. Fourth quadrat, you get nothing. Okay, fifth quadrat, you get 2. So now what is the frequency? Okay, so frequency equals to you see how many quadrat which has the species. Don't add up 2 plus 3 plus 1 plus 0 plus, don't add. You just see how many got. So this one got, this one got, this one got, this one don't have, this one got. Okay, so you can see there are 4 quadrats with the species that you want to study. And altogether there are 5. You throw 5 times. You throw 5 times because 5 quadrat. Okay, you throw 5 times. And out of the 5 times, 4 happen to have the species. You got 1 that don't have. So you don't put that one. You 4 over 5 times 100%. You will get 80%. So your frequency of your this one for your pokok semalu is 80%. That means you throw 100 times, you get 80, or 80 times you will get your plant inside there. Okay, now that's the second one. This is the frequency easier, right? Now the next one, percentage coverage. This is usually the surface covered by the plant, usually for moss, lumut, mosses, or lichen, or things that you cannot separate. Sometimes the let's say the grass, they are all like, you know, like serai. Serai is so difficult to count one pokok because all like clusters, right? So if you want to count in terms of the coverage, you just see how much of the quadrat got the plant cover. So you see here, these are your, let's say you do not want to count one by one, all right? Actually it's better to count one by one with grass. If you can separate out, like I can see 1, 2, 3, 4, 5. Okay, but never mind, you count as coverage. It's about 1/4, right? Out of the whole area, the quadrat, that's why you put small boxes inside there. Out of the 4 boxes, 1 box is already full. It's about 25%. All right. So the percentage of coverage is also in percent. So how to find? Let's say give you example. This one is, let's say, moss kulat, not kulat, lumut, right? Lumut cover on a rock. So the first quadrat, roughly you estimate 0.3. Okay, 0.3 because the whole thing is 1. 0.3 is about 30% covered. The second one is about 50% covered. Okay, 50%, 0.5 meters square. Then this one is more, 0.8 meters square. Then this one 0.4, 0.5. So how do we get this number? It's because we have these boxes, we have smaller boxes inside there, isn't it? So roughly you just estimate how many percent of it got covered. That one, just like when you count your, you estimate the area of an irregular shape, isn't it? So how, if it's inside, you count. Inside the box, more than half inside the box, you count. If it's outside the box, you don't count, isn't it? So something like that. So let's look at percentage of coverage. Okay, now what you need to do is you add up everything, just like the density formula. So 0.3 plus 0.5 plus 0.8 plus 0.4 plus 0.5. Okay, this is the total area, total coverage area of all the quadrat. Okay, then divided by how many quadrat was there? Now total number of quadrat, 1, 2, 3, 4, 5. There are 5 quadrats. Okay, and what is the total area of a quadrat? So total area is 1 by 1 meter, so 1 meter square. Okay, now take note, this one is not always 1. I see students always sometimes make mistake. They always put 1 there. You must see the question. If the question gives you 0.5 here and 0.5 here, this one cannot be 1 already. You see not? This one cannot be 1. This one will be 0.5 times 0.5, which is 0.25, not 2.5. So if you're not sure of a calculation, just put 0.5 times 0.5. Right? This is the area. Understand? Okay. So don't think sometimes students they do calculation too fast. 0.5 times 0.5 is 2.5. Wrong. Please check with calculator. So this will be 0.5. So in this case it's 1. So we put back 1 here. 1 times 1 is 1 meter squared. Okay, so you add up. This is 0.8, 1.6, 1.6 plus 2.5. Yes, 2.5 divided by 5. At the bottom here is meters squared. So, sorry, one more thing. Forgotten your coverage percentage, 100%. Don't forget to times 100%. Forgotten that one. Okay, and then you will have 2.5 divided by 5 times 100%. That will be, you cancel here, you get 0.5, you get 50%. So your answer for coverage is percentage coverage, because the word percentage is always in percent. Okay, so this is how you do. So how you do the calculation. So questions can give any one of these. They can ask you for percentage coverage, usually if it's moss or lichen or any plant that you cannot separate. Okay, and then frequency is easier. The easiest one is frequency. See how many got the species you want divided by the total quadrat times 100%. For density, you count all the number in all the quadrat divided by number of quadrat and also divided by the area per quadrat. Okay, so this is for plants. Right, plants. Now let's look at for animals. Animals, we cannot do quadrat sampling using quadrat. Okay, the reason is because animals can move. Once you put, let's say you throw your quadrat there. When you throw a quadrat there, maybe they got snail or maybe they got ants, right? When you, by the time you walk and you start counting, they run all over the place, isn't it? So you don't know whether to count, want to count or don't want to count because they run out already, isn't it? They will run, isn't it? So you cannot use quadrat sampling for animals to find the population size. You need to use another method we call CMRR. It stands for capture, mark, release, and recapture. Okay, this is interesting. Actually, I like all this ecology. Okay, so how do you do your CMRR method? So this method is used to estimate the population size of animals such as foxes, sharks, actually small animals, snails, millipedes, insects, they can move freely. Now in your textbook, they say quadrat also for small, slow moving one. So we have to follow, right? It seems here, quadrat sampling is used to investigate the distribution of population of animals, right, which are inactive. That means they don't move very much or they move slowly, such as coral and snails. So this is in your textbook. All right, so you take it as it is, right? But usually for questions that come out for quadrat sampling, they give you plants only. Okay, so I would prefer actually when you do your investigation, you do for quadrat sampling for plants, where for animals, it's preferable you use the CMRR. Okay, so now let's go back to CMRR. Now, how do you do that? Right, garden snails, you can use it for garden snail. You can use it for population of wood lice. Okay, the wood, the lice that kutu kayu, that live in the trees. So how do you do it? First of all, you capture, because the word C. C stands for capture. Capture, that means you just put a trap there, at the area that you want to study, and let the animal go inside, because it's a trap. Okay, animals are not that smart, right? So as long as you put food there, they will go in, right? So this is a trapping. First step, you must trap. You trap all the animals that you possibly can, as many as possible, all right? Then you mark all of it. You mark all the animals that you get. Depends on, you get 100, you got to mark 100. You mark it with something, an identification mark that you can recognize later. So you put a ring, okay, or put a tag. If for big animals, you put a tag on the ear, For these cows or anything, and you put a tag on the ear. Horses also, for goats and all, they put a tag on the ear. Or you can put non-toxic and waterproof ink. The ink must be charat, must not be harmful to the organism, because if it's harmful after it dies off, then your population will be affected. Okay, you don't get the animal, you don't get to count the animal later. So non-toxic, and also it has to be waterproof, so that when the rain comes, right, it will not be washed off. Otherwise, it defeats the purpose, right? You put your dot there, you color it, and then later on when the rain comes, then you find, oh, everything all gone, isn't it? You have to redo your first capture again. Okay, so let me check now. Hi, oh, Cikgu Satvinderjit. Hi, okay, good morning. Okay, thanks for joining my class. All right, so you put a ink. Usually we use Indian ink, right, which is waterproof or cellulose paint. They will not be washed off by the rain. Okay, and also the shara also not only that, it must not be able to injure the animal. It's not going to harm the animal. It's not going to interfere with its daily life. That means it's not going to interfere their movement. It's not going to stop them from going into their burrow. It's not going to stop them from going into their cave, right? And you cannot make them so easily seen by the predator. You cannot be putting one big red dot there and make the predator find out, oh, that is an animal that I can see, I can eat. So you're going to harm the animal because you make it easily to be seen by the predator. So you got to choose your marking carefully, all right, to make sure that it does not affect in their daily lives. It's not going to make them more easily to be eaten. It's not going to make them die easier. Okay, so you do that. All right, so you mark all of them. Then after the next stage, you must release. So the first thing is C done. Your M is also done. Mark. Now the third R stands for the third letter R is to release. So you release. Make sure everyone goes away. All right, but you release it in the same place, and you must allow time for them to mix. Then your answer will be more accurate. You must let them time. You do not release and then immediately go and capture balik. Okay, it's wrong because there's no time for it to mingle. When you give it time to mingle, then it's more accurate the answer that you get later. So you give it usually about 2 to 3 days. Okay, 2 to 3 days is good enough time for them to mingle, and maximum 4 days. But don't put it more than 1 week. Okay, because what happens if it's more than 1 week? More than 1 week, a lot of things can happen to the population. Some will die. Some maybe some will die off. That means it will affect the population and become less. Or maybe there's going to be new birth. Some animals are born. Okay, some new animals come or are reborn, and some maybe animals from another habitat come and move inside. So there's immigration, or there could be emigration. Some animals may move away. So in other words, it affects your population size. When you do experiment, you must not let your population size affected. You must assume that when your first capture and your second capture, the population doesn't change. Therefore, you cannot leave it for too long. Okay, so usually 3 to 4 days is good enough for them to mingle. And then what you need to do, you have to trap them again. This is called a recapture. You trap using the same trap, right? And then you put at the same place. You see the second time how many organisms, the animals will go into your trap the second time. Okay, so now you trap it. So after that, you count. Okay, every time you do this sampling, you count. Okay, first capture, how many? Second capture, how many? After that, the second capture, you find that some of them are the unlucky ones. What do I mean by unlucky ones? Because these are the ones that first time capture, you mark them already. Mark them, put a dot there. Okay, put a dot here and a dahi. Okay. Then after that, you release, and then they are stupid enough to walk into your trap again. So they are again, you will see in your second capture also the same animal with a mark. That means second time capture. Okay, they are so unlucky, the 2 times also get captured. And then of course, there are also some which are first time didn't get captured, so they got no mark. So there will be marked and unmarked animals in the second capture. Okay, so now you can ready to put your figures into the formula. You remember this formula? I always remember A times B divided by C. Okay, I always tell my students very easy. A times B divided by C. Now, what is A? What is B? What is C? Okay, very simple. A is your first capture. How many did you get when you trapped the first time? Okay, now look at this example here. A student wanted to estimate the population size of woodlice under a huge tree. He caught 120. Okay, all the figures are given underline, or you just like this one. Okay, or exam don't highlight because when you scan it, your highlighter will cover your answer. You can highlight question, but this is something for me to tell you. Do not highlight your answer with highlighter, because when they scan it, they're going to mark it by scanning. When they scan it, your highlight will become all black and it covers your answers. So please do not highlight your using your highlighter for your answer. You can highlight the question, because question we don't need it, and the examiner not looking for the question, they look at your answer. Okay, so here, let's say I don't highlight, I'll give you a wrong answer. You just circle it. 120. This is your first capture. This is A. Put 120 as your A. Okay, and then mark. They mark each one. They mark 120 using a small dot of cellulose paint. Okay, after that, the woodlice are released. After 2 days, he made a recapture. So this is a second capture. The second capture, 100 are caught. So this is your B. B is the second capture. A is your first capture. Okay. Then among them of your 100, 100, some will be marked, some will be unmarked. The marked ones is the second time they really so unlucky, the 2 times already kena capture. Okay. So the second, the 24 is got marked one. So this is C. C is for those that are marked. Sorry, number of marked animals. Missing one word here. This is a very important word. Marked animals. Okay, marked animals are not unmarked. Unmarked you don't count. Unmarked you don't want. So you put this in your in your formula. A times B divided by C. So 120. So the population size is 120 times, what is your second capture? 100 divided by 24. Okay, so you work with your calculator lah. Alright, I think 500 I don't know. Okay, 120 times 100 divided by 24, you get 500. Now, what is the unit here? The unit here is simply woodlice. This is called individuals. No need to put per area. No need to put per meter square because you're not talking about the area. You just want to count how many individuals. So 500 individuals. That means in estimation, without counting all of them actually, you only catch 120, isn't it? And then by doing this investigation, you found that actually there are about 500 individuals in that area without actually having to count all of them. So this is the sampling. Now, if you want it to be more accurate, what you can do is you repeat the experiment, okay? Or you can catch more than one trap. That means the first capture, you increase your size sampling. That means instead of catching 120, maybe you put 2 traps, you get 240. That's more accurate actually. And you increase your sampling size. This is important. It actually makes your investigation. All right, okay. So let's go on to the last part. Okay, so let's go on to the last part. Now, the following assumption. This is quite of a question they always ask in KBAT. In objective, yes, they will come in objective. What is the assumption that you do when you use your CMRR? What is the assumption? Now, for example, is whatever you do for this CMRR, the ink that you use or whatever you do not harm the animal until the population will become less or you make them die. Okay, so that means the marking must not be hazardous. It must not be poisonous to them. And there's no predator. You must assume that there's no animal eating up the population. There could be but you must assume that the population is stable. So here your assumption here is your population under investigation. The population size is considered stable. Stable, that means you won't increase, decrease. That's why your study, you must not put it for too long. Your first capture and your second capture must be done within 3 to 4 days. Cannot leave it for too long. You leave it 1 month, the population definitely will change. Okay, it's not accurate anymore. So no predator, you must assume there's no predator there. Animal population is stable. Yeah, that's why I must say no significant changes during the investigation. First capture, second capture, you have to assume that no immigration or no emigration. That means no new animal come inside and no animal go outside. Even if there is, the rate must be the same. How many come in is equal to how many they go out. So this is the rate. Okay, immigration and emigration is the same. And the rate of death is the same as rate of birth. That means how many die within the first and the second capture is equal to how many are there born. That means the number is still the same. Okay, you must assume. Mortality rate is the same as death rate. So this is a new word that I want you to understand. And then the next one, you must let them time to mix randomly so that your answer is more accurate. So you let them mix. All right, mix it accurately. Let them mix randomly. And then one more thing, you want your answer to be more accurate. Usually this will ask you. They ask you if it comes out in the structure, they ask you how do you change or manipulate so that this investigation is more accurate. So your answer is usually you increase the sampling size. Increase the sampling size by taking more samples or large samples. So instead of setting up 1 trap, you set 2, 3, 4 traps. And if you put 2, 3, 4 traps, make sure the second capture also use the same number of traps. You cannot reduce the number of traps, otherwise it's different already. So you use 3 traps. The second capture, you also use back the same 3 traps. Okay, so you catch more samples. you put inside your answer there, your formula there, your answer will be more accurate. And then it must be captured at random. That means you need to give time for them to mingle. Otherwise, if you don't give time to mingle, you catch back, more of it is the one that is marked because they got no time to spread out. Okay, you must give them time to spread out. Okay, so I've gone through two methods, two techniques which are important in population ecology. One is for plants, you use quadrat sampling. So you got to remember the formula lah. Okay, how to remember? By doing questions. The more questions you do, you will remember the formula. And for CMRR, it's just A times B divided by C. Now, this I want to tell you, sometimes the question wants to mislead you. If they give you the formula, let's say, first time 120 wood lice were caught, second time 100 were caught, and then among them, let's say 24 were found to be, what happens if they give you unmarked? Okay, now look carefully. They give you 24 are unmarked. So if you put 24 unmarked, can you put your this one here? Can you put your 24 here? Please read carefully. Sometimes they want to test whether you're really, really good and really understand. Unmarked. Unmarked, you don't use it. So how you want to change this calculation? It's not 500 already if it's unmarked. Okay, can I see by what is your mark there? Okay, let me see in the comments. Girls, try to give me your answer. Now for here, I change the question. 24 are found to be unmarked. Okay, 24 are found to be unmarked. So what is the C here? What is the C here? Okay, let me see. Got answer or not? Not difficult. Try to think a little bit. It's not difficult. How can you find the one that are marked? How to find the one that are marked? Okay, any answer? I'm still waiting. Let me check. No answer yet. I got. Let me hang on. 76, 76, 76, 76. Oh, okay, good. Now how you get 76? Because the second sample is 100, right? 100. 100 is the second sample. Out of your second sample, there will be some that are marked and unmarked, isn't it? So 100 is including the marked and unmarked. Okay, unmarked. Yes, unmarked. So if I give you unmarked is 24, then that means the remaining must be marked, isn't it? So I take the 100 minus 24, I will get 76. Okay, so sometimes you have to use your brain a little bit. They try to turn a bit the question by giving you a mark. So be prepared for that. So you don't put 24, you don't see whatever number that you given to you, just simply put, use your brain a bit. They say unmarked is 24, that means you must take the mark 76 and put inside. So your answer will be different. Okay, if it's more of it that is marked that you find it, the answer will be definitely less. Okay, 120 times. So it's going to be 1200, 120 times 100 divided by 76. Now, what answer you're going to get? You're going to get a percent. You're going to get a fraction, right? Now, do I put it in fraction? Do I put it in decimal or not? 157.89 individuals. Does it make sense to have a fraction of an individual? Or do it make sense of a decimal of individual? Usually, for this estimation population, you don't put in fraction, you don't put in what they call decimal, you put it as a whole number. Okay, because it's already estimated, so it doesn't matter. So it could be, your individual will be 158 individuals. Because it's not possible to half individual. You can't have a half an individual. But the density, yes, density you can have decimal. Density here, for this one, density, you can have decimal. So here you can have 3.4. It's perfectly okay because it is a density. It is how many per meter square. Okay? Here, so here per meter square is okay. This is for density, 10 for decimal. Here for estimation of population, you put as a round number, a whole number. Understand? All right. Okay. So any questions from you? Remember to go to Google Meet afterwards. Yeah. Okay. 76. Okay. Very good, girls. You're using your brain, right? So we'll go to Google Meet. All right. And then we do some questions. I want to test whether you can really know how to do this. So this is today's lesson. I just mentioned the population, the technique, sampling techniques. Make sure you remember the formula and how to use it. That's most important. Okay, so I will leave you now. All right, we will go to Google Meet. I will thank you very much for this audience who are following my class. Okay, I will see you again in my next class. All right, I will be consistently doing classes even the holidays. I think Saturday, I'll take 1 hour of the Saturday to go through some of the previous classes. I've had a request from other people. They said they're looking for this topic, they cannot find it. So because I didn't do it, we do it in school already. We already covered in school. So I will do that during my free time. I will have extra classes, so you girls can follow and join and revise your previous lessons. Okay, so I will end my class today now. All right, see you in my next class. All right, thank you everyone. Bye bye. See you.